All right, welcome you guys. Today we're going to be talking about the molecules of cells, and more specifically kind of organic molecules. So organic molecules are really the molecules of cells of life. So we'll be talking about each of those. So we're going to talk a little bit about what are organic compounds or organic molecules, why are they important, and then we're going to specifically go through what are carbohydrates, what are lipids, and what are proteins, and what are nucleic acids. So these are our organic compounds or organic molecules of life. So what are organic compounds? So if we remember last week when we were talking about the different elements, and we had some elements that make up all life, right? And so carbon and hydrogen are two of those elements that are in all of life. And so organic compounds have to contain carbon and hydrogen, and they're really the building blocks of life. And what's special about carbon is that it can actually have up to 4 covalent bonds. So remember we were talking about our different types of bonds, and covalent bonds are where the two atoms share electrons, right? So if we have a carbon compound here, so we have a carbon atom here, and it can share up to 4 different covalent bonds, okay? And it can be either one double bond and two singles, four singles, two doubles, or even a triple bond and a single bond. And then this just kind of shows the different ways that hydrogen can covalently bond with carbon. And so we can create these different carbon and hydrogen compounds, which are considered organic compounds. Okay. So the size and the shape of an organic molecule really depends on that carbon skeleton. So what do we mean by a carbon skeleton? That's, how many carbons are attached to each other, what shape are they in? So like on our last slide, we have some that were just in a row, but then we also have these ring structures as well. So each corner here is a carbon. So that carbon, like we said, attaches to hydrogen, but it can also attach to other things such as oxygen. Okay, and these different guys that are hanging off or attached to the carbon, or this carbon skeleton we call it, these guys are called functional groups. So if it's anything other than just a single hydrogen, such as an oxygen and a hydrogen, or the CH2OH, these are called functional groups. And the functional groups are what give the compound its specific specialties. So such as glucose, okay, so the functional groups on glucose are going to make the molecule hydrophilic, meaning it loves water, or it dissolves in water. And we talked about water as being a great solvent, right, in our water lecture. So these functional groups are what give these different compounds its special functions, okay? So what are some common functional groups that we talk about in organic molecules? And the one I really want you to know is the hydroxyl group. Okay, so that's that OH that we just looked at on the glucose. So that is something that's going to cause that compound to be hydrophilic. And we said hydrophilic means that we like water. So anything philic means you like it, phobic means you dislike it, and then hydro is water. So hydrophilic, we like water. Okay, so for example, this is alcohol, the compound alcohol, and so because it has that OH hydroxyl group, alcohol dissolves in water. Okay, and so there's these other functional groups as well, such as a carbonyl group, which is a double bound carbon to an oxygen, or you can have a carboxyl group, which has that double bound oxygen and an OH. So again, you just have these different functional groups that gives that compound specific properties, okay? And that being hydrophilic or hydrophobic is one of the properties that a compound can have. So now if we look at organic compounds that come together, so we can have smaller compounds and bigger compounds. So when we're talking about cells themselves, which is the smallest unit of life, they're going to actually be making these macromolecules, which are larger molecules. And essentially they put together these smaller molecules into bigger molecules. So what are the names of these different size molecules? When we're talking about these small building blocks, we call them monomers. So mono kind of meaning one, so just kind of one building block. And then you put multiple monomers together to create a polymer, which would be a long chain of monomers strung together. So for example, if we're talking about glucose, this glucose molecule over here is just one monomer. And then to make up starch, which we should be familiar with starch, it's the stuff that's in like potatoes and grains and things like that. It's essentially a long chain of glucose molecules. So glucose monomers all strung together to create a long polymer called starch. Okay. So how do we actually build a polymer? Okay, so we take our small monomers and we build it and create a long polymer. But how does the environment or how do cells actually do this? Well, it's called dehydration synthesis. So what actually happens? Well, what we do is we remove water from two molecules so that they bind together. So for example, we have, so each of these little purple dots is a carbon compound, okay? So we have that hydroxyl group, the OH, on one compound, and then we have just a single H on another compound. So we're going to create one larger compound by taking water out. So we combine the two H's, and then that's H2O with the oxygen. Okay, so dehydration synthesis, think you're dehydrating it, so you're removing water. Okay, so that's what's happening with the dehydration synthesis to create a larger polymer or a larger molecule. So for example, in the environment, when we're talking about plants, they're actually able to use photosynthesis to make glucose. So then they can store glucose in some specialized cells as starch. So essentially they are creating starch out of those glucose molecules by removing the water and creating this dehydration synthesis. Okay. So now we know how to build a polymer. So we remove the water and put those two compounds together to create a polymer. How do we actually break down a polymer? So if you're given a large compound such as starch, how do we break it down into glucose? So this process is called hydrolysis. So we actually break down those polymers with water. So now we're going to add water. So in our dehydration synthesis, we're removing water, and in hydrolysis, you're adding water. Okay. So if we take that compound we just made in the last slide and we add H2O, well, the H is going to add to one compound and the hydroxyl group or the OH is going to add to the other compound. So then we break them apart. And we're doing this every day. So if you eat something with starch in it, such as a potato, the enzymes in your digestive tract are going to be able to break it down into monomers. And we add water to that. So the enzyme uses water to be able to break down those polymers of starch. So now you can stop the video for a second or pause the video and test yourself and say, is this reaction hydrolysis or dehydration? Are we adding water or are we taking away water? So hopefully you were able to pause the video and answer this for yourself. So if we have these two compounds here, such as glucose and fructose, and we want to add them together, okay, so we're creating a larger compound or a polymer. And what happens is we're going to take away water. So we're going to take away this OH and H to create water and sucrose. Okay, so fructose and glucose together make sucrose. And so that would be dehydration synthesis. Okay. So now that we know what organic compounds are, how do we make them, how do we break them down, let's talk about the specific organic compounds that are in our body and in our cells. So the first group we're going to talk about are carbohydrates. And all carbohydrates contain a very consistent ratio. And this ratio is if we have one carbon, we're going to have two hydrogens and one oxygen. So it's a 1-to-1 ratio. Okay, so we can build bigger carbohydrates, but they usually are going to contain that same ratio of elements. Okay, so most carbohydrates, or all carbohydrates, are hydrophilic, meaning they like water because they have that hydroxyl group we talked about. So that OH functional group is going to make them hydrophilic. Okay. So the first group of carbohydrates we're going to talk about are called monosaccharides. So monosaccharides are simple sugars. So they're going to be those monomers, those one compound molecules, and we're going to build other things from our monomers, our monosaccharides. Okay, and so our 5 different monosaccharides that we have are glucose, fructose, and galactose, and each of them have 6 carbons. So if we kind of put that into our ratio, if we have 6 carbons here, that means that we're going to have twice as many hydrogens. So that's going to be 12 hydrogens, and we have the same amount of oxygen. So then we'll have 6 oxygens. So it'll be a 6, 12, 6 compound, okay, for all these monosaccharides. And then we have 2 more monosaccharides called deoxyribose and ribose, and they have 5 carbons. Okay, so if we put those 5 carbons back into our ratio, if we have 5 carbons, we're going to have 10 hydrogens and 5 oxygens. So 5, 10, 5. Okay. So if we look, here's our glucose on the left. So we have 1, 2, 3, 4, 5, 6 carbons, and we have, well, you can count them, but we don't have to count all the hydrogens and oxygens. But the important part is we have these hydroxyl groups, right? These functional groups that are going to make glucose hydrophilic, okay? And then we have fructose, which is similar to glucose, but if you see that that double bonded oxygen is in a different location, so it's on the second carbon instead of the first carbon. So again, they're similar and they have the same amount of carbon, hydrogen, and oxygen. They're just slightly different in their structure. Okay? So if we look at some more carbohydrates, we had our first group that are monosaccharides, and if you put 2 monosaccharides together, then we get our disaccharides. So di meaning 2, right? Mono meaning 1. So if we put 2 monosaccharides together, we're going to get these 3 different disaccharides. So maltose is composed of 2 glucoses. So if we do our dehydration synthesis and stick 2 glucoses together, remove water, we'll get maltose. Okay? And then sucrose is composed of 1 glucose and 1 fructose. Okay, and sucrose is what you know of as sugar, right? And then lactose is composed of one glucose and one galactose. Okay, and that's what we now think of as milk, or what is in milk, right? It's the sugar compound in milk. And it's also what people are allergic to when they say that they are lactose intolerant, right? Meaning they lack the enzyme to break down lactose into glucose and galactose. And we'll talk a little bit more about that, okay? And I have a fun video on why people are actually, intolerant to lactose, okay? So you can pause the video now and let's review. If we were to break down lactose, what process would we use? So we just kind of talked about how we build them up. How do we build a disaccharide? We put two monosaccharides together, right? So they're each composed of two monosaccharides. So if we were to break down lactose to get glucose and galactose, which process? Think back to how do we build, how do we break it down? Which process would we use? So hopefully you paused the video and were able to answer that question. And it would be hydrolysis. So hydrolysis is how we break down our larger compounds, such as a disaccharide, into monosaccharides. So we would add water, right? So add water to break down lactose into glucose and galactose. So now our last group of carbohydrates are called polysaccharides. Okay, so polysaccharides are macromolecules, so they're really large, and they're composed of thousands of monosaccharides linked together. And how do we link them together? We dehydrate them, right? So we take away water and we link them all together in dehydration synthesis. So this is our example as starch. So we have a couple different polysaccharides, and our first one is starch. And starch is a way that plants can store sugar. Okay, so they put glucose monomers all together into a chain, and that chain is a starch molecule. Okay, so it's how plants store their energy, and then we eat that energy and break it down. Okay, we eat that starch, we break it down into our monomers such as glucose. Okay. So all these plants that are grains or vegetables such as potatoes, any tuber, right, that kind of lives in the ground, like a carrot, potato, sweet potato, all those guys that are in the ground, they hold that starch in the potato, right, or in the grain, right? And then, so we break it down and we use that as energy. Okay, but that's how the plant itself stores its own energy as well. So our next polysaccharide is glycogen. Okay, so glycogen is, again, it's going to be a long chain of glucose, but essentially it's a branched chain. So if we looked at starch, it was just one long chain, and then now glycogen has multiple branches on that chain. Okay, so it's a slightly different type of compound because it's branched. Still, it's glucose monomers, right, put together to create this large polysaccharide. And this is how animals store glucose, but it's short-term storage. So you would find this in muscle cells or in the liver cells. Okay, so this is how we store glucose for short-term energy use. Okay, we store it long-term as fat, and we'll talk about that in a little bit. So another polysaccharide is cellulose. So cellulose is actually used as a structural component of the cell wall of plants. So again, it's a chain of glucose monomers, but they're stuck together slightly different. So every other glucose is flipped upside down. Okay, so it's a slightly different connection between the glucose molecules. So that's why it's different from starch. So it's very abundant in those cell walls of plants. Okay. Now we have another polysaccharide that many people don't even think about or know about, and that's chitin. Okay. And it's a structural polysaccharide as well, just like cellulose is a structural component for plant cells. This is in the exoskeleton of insects, crustaceans, and even in fungus, like mushrooms, right, in the cell walls of fungi. So it's a structural compound as well, just like cellulose, but it's even stronger, right? So very strong structural component for these insects and crustaceans. So let's pause again, and you can review a little bit so you can name some different carbohydrates. So name a few monosaccharides, a few disaccharides, and polysaccharides. Okay? So hopefully you were able to pause the video and maybe write down a couple examples of each of these. So if we look back at our monosaccharides, we were talking about glucose, fructose, right? And then we put those monosaccharides together to make our disaccharides, such as sucrose and lactose, right? And then some of our polysaccharides, some examples, we just talked about chitin, but also starch, glycogen, and cellulose, okay? So now we're going to switch gears a little bit and start talking about lipids. So carbohydrates, you kind of think sugars, right? Starches. So lipids are fats. And so just like carbohydrates are hydrophilic, meaning they like water, they dissolve in water, lipids are hydrophobic, meaning they do not like water, they do not mix with water. And so if any of you have ever either used an oily salad dressing or tried to mix oil and water, you know that they completely separate, right? The lipid or the fatty oil just sits right on top of the water. So they are hydrophobic, which is different from carbohydrates. So these guys are the way we store energy long term in animals. So humans and animals, when we eat too much glucose or too much sugar, then we use it, so we store it short term as glycogen, right? But then if we eat more than that, then we have to have another way to store it. And that's how we store it as fat, as lipids. So then we can break it down later if we need that energy. So fats are composed of glycerol and fatty acids. So if we look at our picture over here, we have this glycerol head, they call it. Okay. So the glycerol then is attached to 3 chains of fatty acids. Okay. So we'll look at these different types of fatty acids and why they are important. So if we take those fats, we can separate them into 2 categories, okay? So we can call them saturated or unsaturated. So I know many of you have probably heard, oh, don't eat those saturated fats, right? They're bad for you. So, why are they bad for you? And what's the difference? What makes something saturated versus unsaturated? So saturated fats are fatty acids with just a single bond in the carbon chain. Okay, so they are essentially kind of these straight fatty acids. right? So they're single bonded, they can kind of stack together, and the saturated part means that they're attached to as many hydrogens as possible. So every carbon is attached to as many hydrogens as possible. So they're saturated with hydrogen, is where the terminology comes from. And a saturated fat is solid at room temperature. So you think of animal fats, right? Butter. So they're a solid at room temperature. Okay. Now, unsaturated fats, the fatty acids have some double bonds in there. So essentially the double bond creates a kink in that chain. Okay, so they're not these nice straight chains of fatty acids, they have kinks. And they're not saturated with hydrogen. So because it's double bonded, that carbon is double bonded to another carbon, so it doesn't have as many hydrogens as it possibly can. And in general, unsaturated fats are liquid at room temperature. So you think of kind of your oils, right? Your olive oils, okay? So now we know that fats are lipids, but we do have a couple other types of lipids which are not saturated or unsaturated fats, but they are very important in our bodies. So another kind of lipid is a phospholipid. And essentially a phospholipid has that glycerol and only 2 fatty acids. So instead of 3 fatty acids, like our saturated and unsaturated fats, it only has the 2. Okay, and the other big thing is it has this phosphate group up here attached to the glycerol. Okay, so it's a little bit different than our other lipids that we were talking about, because we add a phosphate group and we take away one fatty acid chain, because that carbon that was attached to the fatty acid is now attached to a phosphate group. And what happens and why these guys are important is that now, because we've added this phosphate group, we've created a hydrophilic head. So that means that this head of the phospholipid likes water, but the tail does not. Okay, so the tail is hydrophobic still, they're fatty acids. And these guys, these phospholipids, are really important in the cell membranes. So the phospholipids create the cell membranes, and we'll talk a little bit more about that when we talk about cells and things, but phospholipids create our cell membranes. So another type of lipid that you may not think about, of course we think are fats, right? So fats, saturated, unsaturated. Now we just learned about phospholipids, which are really important in the cell membrane. And now we have steroids. So steroids are hormones, right? So hormones, we have different types of hormones in the body, but we do have steroid hormones. And steroids are a little bit different because they're essentially a couple of carbon rings fused together with different functional groups. Okay, so instead of like a chain of glucose molecules we were talking about in carbohydrates, these guys are stuck together in a different way. And we may have 6 carbons, we may have 5 carbons, and we have these different functional groups. And you don't need to know the specific structure or anything like that, but just know that steroids are really important in our body. So of course our sex hormones are made of steroids, but also cholesterol. So cholesterol is really important in our body. It is a lipid, right? And it's specifically a steroid lipid. They're also very important in the membrane of the cells as well. So we said those phospholipids make up the majority of the membrane, but stuck in the membrane of the cells, or in the cell membrane, are these cholesterol molecules. And again, we'll talk a little bit more about cells and the cell membrane when we get to that chapter. Okay, but just think steroids are a form of lipid. Okay. So go ahead and pause the video again for a little bit of review so you can go back through and name our 3 different types of lipids. What do they all have in common? But what are some differences between maybe the saturated and unsaturated fats? Okay. Okay, so hopefully you were able to do a little bit of review and pause the video and answer these questions. So what are our 3 types of lipids? So we have fats, right? Saturated, unsaturated. We have phospholipids and we have steroids. Okay, so those are our 3 types of lipids. What do they all have in common? Well, they're all hydrophobic, right? So they all don't like water. And what's the difference between a saturated and unsaturated fat? What did we say was that difference in those fatty acid chains? Saturated, we have no double bonds. It's all single bonds saturated with hydrogen, so they're nice straight lines. Whereas unsaturated fats have some double bonds in there, so they have some kinks, right? Some bends in there, so we don't have those nice straight saturated fats, or sorry, fatty acids, okay? So now another group of organic compounds are proteins. So proteins have a huge range of function and structure, okay? But the common thing of all proteins is that they are composed of amino acids. So amino acids are the monomers, just like we talked about glucose being a monomer of a starch polymer. Well, the proteins are these big macromolecules or polymers made up of many thousands even of amino acids. And they all have different structures, different functions, and our cells are constantly making proteins in the body. And so this is just the standard form of an amino acid. Okay, so all amino acids have an amino group, okay, and a carboxyl group. Okay, so those are those 2 functional groups on an amino acid. And then the thing that changes and why all the different amino acids are different from each other and have different properties is this R group. So this R functional group changes between each 20 amino acid. Okay. So what are some functions of proteins, right? We have so many proteins in the body and they have such a wide range of function. So we have structural proteins, such as in our hair and our nails. We have proteins in our muscles that allow for movement, right? In our immune system, we have antibodies that help us to defend our body against any sort of microorganism. We have albumin in the blood, which is a protein that's helped to store things and kind of takes things through the blood like a messenger molecule, as well as insulin. So hormones are also proteins. Okay, so signaling proteins, which we have steroid hormones we talked about, but we also have protein hormones. So hormones are really kind of a signaling system of the body. Okay. And a lot of our proteins are enzymes, and some of the biggest proteins are enzymes. So enzymes help to break down molecules or build them. So enzymes are kind of the powerhouse workhorses of the body. Okay, making chemical reactions happen faster in the body. Okay, so amylase is an enzyme in your saliva, right, to help break down starch in your mouth. As you eat food, you're already starting to break it down in your mouth. Okay. So we said that those R groups are really determining the property of that amino acid, right? So we said there's a lot of different chemical natures to these different compounds. We already talked about being hydrophilic or hydrophobic, but you can be acidic, you could be basic, polar, non-polar. So that's what that R functional group determines, right? So such as this amino acid, leucine, is hydrophobic, okay, because of that R functional group, whereas these two, serine and aspartic acid, are hydrophilic. Okay? So it just depends on that R group, and it gives it its specific chemical properties. So proteins are made up of those amino acids, right? So we said the amino acids are the monomers, and we bring those together and we form what are called peptide bonds, okay? And a peptide bond is just a very specific covalent bond, okay? So again, it's very similar. We do kind of a dehydration synthesis. And so we remove water from the carboxyl group and the amino group. So we steal that OH and H, we join them together. So this is a very specific peptide bond joining that nitrogen and the carbon. Okay. So then it becomes what's called as a dipeptide. Okay, so we have two peptides, so one amino acid is one peptide, one monomer, and then we join them together and now we become a dipeptide. And you can just create these large macromolecule structures and they're proteins. Okay. So again, it's a dehydration reaction. We're removing the water, so very similar to our carbohydrates we already talked about, right? So now let's talk a little bit more about enzymes. So these are these huge complex proteins, and they're going to regulate chemical reactions. Okay, so we said they could speed up reactions. They really help the body with complex chemical reactions. And these guys are polypeptides. So again, with proteins, just kind of change your terminology a little bit. We've talked about monomers becoming polymers, right? So multiple monomers making up a polymer. Now we're just talking about multiple peptides coming together to make a polypeptide. And we saw the dipeptide in the last slide, right? So just replace, mer, M-E-R, with peptide when we're talking about proteins. So they can build molecules, they can break down molecules. It just depends, okay, what the enzyme's job is. So an example, so these pictures are lysozymes, okay. So there's just two different ways of depicting proteins. So you can have this kind of bubble form or ribbon form. So it's the same protein or the same enzyme, just a different style of depicting it. So another thing about proteins is that they have a very specific shape. So the way that the peptides come together, it's called folding. So they fold into these kind of spiral-like compounds, and we'll talk a little bit more about that. But they can only be active if they have that very specific shape. So you have a coil here and a coil here, and you have the active site of where that enzyme may function, what it binds to. And so if you put that protein in some sort of environment that it doesn't like excessive heat, right? Say you heat it up, that protein structure is going to unravel. So it's going to lose its shape. Therefore, it's no longer a functional protein. So if it's an enzyme, it no longer can bind to the chemical and help that reaction. Okay. So we call this process denaturing. Okay, so you can denature proteins by putting them in unsuitable environments, such as excessive heat. And that's part of what happens when we cook our food as well. So we're kind of denaturing some proteins in there. So I said that proteins have to be built a certain way, folded a certain way, so their structure is very important. So there are 4 levels of structure to a protein. And what happens is they use hydrogen bonds to fold. Okay, so let's look at these 4 levels. So you have your primary structure, which essentially is just going to be the chain of amino acids, right? So we did our dehydration synthesis. We have some peptide bonds here. So we've just got a chain of all these amino acids. Well, now we have a secondary structure. Once you have that chain built, we can start folding. And we can fold it either in a helical shape, meaning a spiral, or in a sheet, so a pleated sheet. So it kind of bends and folds like an accordion. Okay, so you either have this beta pleated sheet folding or this alpha helix folding and bending. Now, when you put those guys together, you can have both forms of the secondary structure into a tertiary structure. And now you've created a polypeptide, right? So now you can even put multiple polypeptides together, and now you get a quaternary structure. And that is what a protein is. So a protein is multiple polypeptides linked together to create a protein. So there are these huge structures full of many, many, many amino acids, okay? And so this structure, like I said, is very important for the function of the protein. So to be an active protein, you have to be folded and in this very specific structure to each protein. So let's go ahead and pause again and do a little bit of review on our proteins. So what are the monomers of proteins? What are they made up of? And what does it mean when an enzyme is denatured? Okay. So hopefully you paused the video and were able to come up with some answers. So what are the monomers of proteins? We call them peptides, right? So those are our amino acids. So amino acids are peptides, and we bind some multiple amino acids or multiple peptides together to form these polypeptides. And we put those polypeptides together to create proteins. Okay? So what does it mean when we say an enzyme is denatured? So an enzyme is a type of protein, right, that regulates chemical reactions in the body. But if we denature it, that means that we've put it in some sort of unsuitable environment, such as excessive heat, and we have unfolded it. Okay, so it's no longer has its active structure, and it is no longer an active protein. It is denatured. So our last organic compound is nucleic acid. So we have 2 types of nucleic acids, and they are DNA and RNA. So let's review about kind of what DNA and RNA are. So when we talk about DNA, they make up our genes, right? So our genes are made up of the DNA, and what happens is our body reads the DNA, and essentially it tells you what amino acids to put in that sequence when we're building our proteins, okay? So DNA is deoxyribonucleic acid, and it's a polymer. Okay, so it's a larger molecule. So DNA, right, is what makes up our genes. And then we're going to read that DNA into RNA. So RNA and DNA are our 2 nucleic acids. And then the RNA is what's going to tell us that sequence of amino acids in the protein. Okay, so DNA, we transcribe it to RNA. These guys are our 2 nucleic acids. And then we're going to read that RNA into a sequence of amino acids that's going to become the protein. Okay? So now the RNA is ribonucleic acid. So essentially we just get rid of the the deoxy, right? So it's just ribonucleic acid. So these guys are both our nucleic acids. So we only have 2 nucleic acids. So DNA and RNA are polymers of nucleotides. So DNA and RNA are nucleic acids, and just like amino acids make up our proteins, nucleotides make up our nucleic acids, okay? And all nucleotides consist of 3 parts, just like our amino acids have 3 parts, okay? So we have a sugar, which is either one of 2 sugars, and we've talked about these sugars before. These are monomers, monosaccharides, so deoxyribose and ribose. Okay, so that's our sugar, so it's going to be one of the 2. If you're DNA, you have a deoxyribose sugar. If you're RNA, you have a ribose sugar. So that's what determines whether you're DNA or RNA. Okay. Then you have a phosphate group, okay, and a nitrogen base. Okay. So what are our 4 different nucleotides? Okay. So we have adenine, which we term as an A, thymine, which is a T, cytosine, which is a C, and guanine, which is a G. And the nitrogen base is what determines which nucleotide it is, okay? So if it has an adenine base, it's adenine, and so on, right? So that's what determines which nucleotide it is, okay? Is that base. So now when we're talking about RNA, okay, RNA has the same 3 bases, same nucleotide bases, A, C, and G, but instead of thymine, it has a uracil. So then we have a U. So anywhere you see a T, it'll be a U instead. So RNA is very similar, but it has a U instead of a T for the nucleotides, okay? And it has the ribose sugar instead of the deoxyribose sugar, okay? So when we build our DNA, right, it's gonna fold very similarly to protein, but in a very specific way. So whereas proteins can fold in kind of all these different ways, it can form helices or the beta pleated sheets. These nucleic acids only fold in one specific way, and that's a double helix form. Okay, so instead of just an alpha helix, you have a double helix. So this is your classic DNA, right? So most people are probably familiar with the structure of DNA and that double helix look, okay? So essentially what it is it's composed of 2 polymers of nucleotides. So we have a string of nucleotides on one side and then another string of nucleotides on the other, okay, running in the opposite direction. And so we call those anti-parallel strands. So one is running in one direction, one is running in the other direction. And what happens is that those nitrogen bases line up to each other, and so they can form a hydrogen bond to each other. So the nucleotides are going to have hydrogen bonds, and they bind in a very specific way. So only A binds to a T, and only a C binds to a G. Okay. Now, if we're talking about RNA, an A lines up with a U. So I said just replace all T's with a U, right? So those are our 2 base pairs, right? So our 2 types of base pairs, we can have an A and a T or a G and a C, and that's it. okay, which is why it creates this very specific double helix pattern. Okay. So let's pause the video again and do our last review. So what is the connection between nucleic acids and proteins? We've talked a little bit about their similarities and differences. How many nucleotides are there in DNA and RNA? And then how many amino acids are there in proteins? Okay. So hopefully you're able to answer these guys. So what is the connection between nucleic acids and proteins? They have some similarities, right? But essentially how they're very connected is that nucleic acids, or DNA and RNA, essentially have the blueprints for building proteins in the body. So we need those nucleic acids, DNA and RNA, to tell our body what proteins to make. Okay. So how many nucleotides are in DNA and RNA? What are those nucleotides? So we said nucleotides make up DNA and RNA, right? And those bases. So we have 4. Right? 4 in each, but we replace 1 when we're talking about RNA instead of DNA, right? So A connects to T, C connects to G, okay, in DNA. Then we replace the T with a U, okay? So those are our nucleotides. And we have 20 amino acids in our proteins. So the proteins are made up of just a different combination of those 20 different amino acids. So we have a lot more amino acids than we do nucleotides. Okay. So now just to finish up with some kind of interesting thing about lactose intolerance. So now that we know what lactose is, it's a disaccharide that's found in milk. And actually, lactose intolerance is the norm, meaning that all animals, including humans, become lactose intolerant when they grow up. Right? So really only growing animals should be drinking the mother's milk. But then once they're adults, they should become lactose intolerant. But it's actually really interesting because humans have developed a mutation in our genome that allows us to be lactose tolerant, meaning that we keep the enzyme around in our body to break down lactose. So if we look at our picture here, if we have our lactose disaccharide, and that lactase is the enzyme that breaks down lactose into glucose and galactose. Okay, so essentially our ancestors, they should have lost the ability to break down lactose. But as cattle became domesticated, lactose tolerance people increased. So again, it's that natural selection, right? So, it became an advantage if you were lactose tolerant. And there's actually 4 different mutations in different ethnicities going back, 9,000 years in Europe. Because it became a survival advantage if you were able to drink the milk of these domesticated animals, right? So it kind of came hand in hand, the evolution with the domestication of cattle, then we became more lactose tolerant. Just kind of interesting, right? If you think about it, we should all be lactose intolerant. So that's actually the norm. And anyone that is lactose tolerant has a mutation. So that's kind of interesting and fun. All right, if you have any questions, let me know. Otherwise, hopefully I'll see you guys in office hours. Thanks a lot.